Hello to all of you. We're going to go on to part 4 of endocrine, where I introduce the 3 major categories of hormones, but we're going to start with peptides. So these 3 major categories are peptides, steroids, and amines. So all hormones will belong to one of those classes. But really most of them are peptides, and that's what we're going to start talking about right now. So peptide hormones are water-soluble proteins. It's very important that you remember that they're water-soluble. And I'll tell you why in a minute. They're variable in size. So this picture that I'm showing you here is of amino acids all in a row. And these amino acids will form proteins. And they can be variable in size. You can have peptide hormones of hundreds of amino acids up to thousands of amino acids. And they can be branched out here also. It's not just one single row of these amino acids. So you'll know that if a hormone is not a steroid and it's not an amine, then by default it's going to be a peptide. And that's where the majority of them are going to be in that category. So given that they're water soluble, it means that they cannot cross the membrane, the cell membrane. So when they're bringing a message, they're going to have to stay outside and pass that message on. So how is it, I'm asking here, that these peptide hormones can interact with other cells if they can't even cross the membrane? So they're going to use the receptor, and you've seen that when we talked about insulin, a membrane receptor, signal transduction, and second messengers. Okay, the receptor, the signal transduction, and second messengers. An example of that is oxytocin, but let's just go and see what this all means. Signal transduction, if I had to translate that, is the process of transferring a signal throughout an organism. And especially, we're going to talk about it in terms of coming across a cell. Okay, so a signal is being sent. So in this diagram, you have a hormone. I'm going to show you the hormone there, and it's going to arrive to a particular receptor. And remember, it can't just go barreling on in because this membrane is a lipid bilayer, and this is a water-soluble hormone, so it can't come inside to the cytoplasm of the cell. So what needs to happen is, 1, it needs to interact with the receptor. 2, there's got to be some sort of translation of that signal. And that translation is what I'm calling the signal transduction. It's transferring that signal inside to the cell. For what? For the cell to change its behavior. So a series of steps is going to make up that signal transduction. And the result, the end result, is that there's going to be a change in the behavior of that cell. It's going to do something different than it did before the hormone arrived there. The second messenger part of this mystery are molecules that are going to relay signals received at the receptor on the cell surface. Remember, this hormone can't come in. Such as the arrival of a protein hormone, a growth factor, and it's going to make that change happen inside the cell. I'm directing you to the Amoeba Sisters video in section 1.2 on Canvas. There's a really nice video on second messengers and how they behave. Okay, let's go back to this diagram. We looked at this diagram before when we were looking at homeostasis, and the fish tank was the analogy, where we had a stimulus and a sensor and an integrating center. The way that these worked were the stimulus was low temperature. The water in that tank was too low. And then we had a sensor, and the sensor in the tank was the thermometer. And it detected that that temperature was too low. Then we had an integrating center that was the black box that was going to tell the effector, the target, that it had to turn on the heater. And then the heater turned on And the response that we had was that the temperature went back up. So stimulus, temperature too low. And we ended up through a series of steps correcting the initial problem. So now let's look at that in terms of hormones. It's the same thing. We have a stimulus, which is the first messenger or the actual hormone. It's arriving to this purple area is the membrane, and there's going to be a receptor. So the receptor is in the membrane. It serves as a transducer. It's going to translate the message coming from this molecule that cannot enter the cell and send some sort of signal to the inside, expecting a response. So what happens is this membrane receptor protein is going to activate some other intracellular signal molecule. So we're going to call all of that a second messenger system. So those are sitting there waiting, that signal molecule, and it's going to trigger, it's going to alter a protein that was already sitting there, that was waiting. So it targets that protein. That protein is altered. It changes a little bit. That molecule becomes slightly different, and it's going to cause a response. So we went from having a messenger that didn't come inside the cell, but it was able to send a message, modify a protein, and create a response. Looking at it this way, a signal transduction, we could pretend this is my hormone. Hormone is this first messenger. He's coming with a message. Okay, an analogy. When we talk about signal transduction, we're talking about conversion of a signal. We're modifying a signal some way. So here comes the first messenger, our little guy here with the directions. And those directions are going to have to be translated into some sort of result. So the directions are read. A plan is followed. So this receptor then binds to the hormone. We're going to call this guy the first messenger. And it's going to generate some sort of result. And those results are here. So I want to show you how this cell, this hormone here, is arriving at that receptor, and then that receptor is going to change something. So I'm going to backtrack to that slide of the microphone and tell you that these two are transducers. It's a type of transducer that you know already, a microphone. A microphone converts acoustic energy to an electrical signal, so it's a transducer. It changes one message into a different form. A loudspeaker does the opposite. So it gets that electrical signal and puts it into an acoustic one. So those are transducers. So you know that terminology. So this diagram is showing the hormone again arriving at the receptor that then triggers some sort of cell response. And this is the way peptides work. Never entering the cell itself, but just interacting here with the receptor. The other type of hormones will do it differently. So for now, let's just stick with the peptides that have a signal that is only going to attach on to that receptor. Most signal transduction uses something called a G protein. And what's that? A G protein is like a switch, and it's going to connect to that receptor. So in this diagram, let's look at it. You have your signal molecule, which is your hormone, and a receptor. It's going to engage here. And now these G proteins will cross the membrane 7 times, and they're all part of this G protein coupled to this receptor. So all together, the G protein and the receptor are sort of one thing. And it's a weird thing that it comes in and goes back out and comes in and goes back out. It's just crosses the membrane 7 times, which is a really strange thing. But that's what a G protein is. So what next? I'm still using that same sequence of the stimulus, the sensor, the integrating center, the effector, and the response, right, that we had with the fish tank. So I want to go down this side. There's 2 mechanisms, 1 and 2, but I want to go down this one first. So here's the hormone, attaches onto the receptor. This is the inside of the cell over here. Receptor then communicates with this G protein. It's going to open channels on the membrane. And then it's going to also trigger a response by AE here, which is an amplifying enzyme. Why do we need that? Well, because hormones come in teeny quantities, and they're so small that we need to amplify that response for it to function inside the cell. So think of it as an enzyme, which is something that speeds up a reaction, and it's an amplifier because we're going to have to increase the volume of that response. Think of it that way. Then it'll come, that mechanism here is going down this cascade. So this cascade will now hit the second messenger system, is the integrating center. And it will phosphorylate a protein. So what happens here is that it's going to add a phosphate. When I click this, sometimes it changes. I'm sorry. So a phosphate is a phosphorus with its oxygens. One of them is a double bond. So this phosphate here is going to attach onto a protein. So you've now changed the protein somehow. That change in that protein is going to now trigger a response that it couldn't do before, because this protein was sitting here without that extra phosphate. So if I show it to you backwards, let's do it that way. I want this response for the cell to do something. And how am I going to do it? I have a protein there, but it's not effective yet because it doesn't have the phosphate. Well, how am I going to allow that phosphate to happen? Well, you need the second messenger to be triggered. Well, how do you trigger the second messenger? Well, it needs to be in connection with this enzyme that can only be stimulated had that hormone arrived. So there we did a little piece of it backwards. So these second messenger systems are going to be different for the different hormones. So I'm talking in very generic terms. A second messenger is what? It's just some sort of molecule that is sitting there and will only respond once this G protein and amplifying enzyme are essentially triggered. And they're triggered because the hormone landed in the receptor. So it's the second messenger then that can change something in the cell. What's it going to change? It's got that phosphorylation. It's going to attach onto the protein, and you're going to have a cell response. Okay, this is how this group of hormones works. Remember, this slide is here just to remind you of this feedback loop and how they're similar to feedback loops when we speak of hormones. This is written out for you so that you can go back and see it. The second messenger adds a phosphate. I'm calling it a waiting protein because the protein was sitting there waiting for that phosphate that then triggers that needed response. It's kind of like a tag team going on. So they're my little stick guys showing the tag team that happens when this hormone lands in its receptor. There's a second path that this can go down. It's the second piece here that I sort of ignored at first. We went down this road. Let's go down this one now, where you have a different hormone on a different receptor, and it's coupled to tyrosine kinase, so another enzyme. And these are models. It shows a little spring. It's not really a spring, but it's coupled to this. So once the hormone, first messenger, attaches onto the receptor, it will trigger this enzyme that will then also phosphorylate a protein and create a cell response. Two different mechanisms, but they are very similar in that the hormone is not crossing the membrane. A little more on tyrosine kinase. It's a receptor enzyme. It's actually 2 in 1. Hormone binds to the receptor. The enzyme is activated. Transfers that phosphate group from ATP. Where is it going to get the phosphate from? It's getting it from ATP in the cell, attaching that phosphate onto the protein, and it changes the protein's shape because now I have a phosphate attached onto that. It's going to cause some sort of response. So we want to think about how long does it take until this can happen, until you get a response, and from the time that the hormone attaches until you get a response. And how long is it active for? So it works fast because second messengers modify existing proteins. That protein was already there. I don't have to go manufacture a protein. It's waiting. It's sitting there. And all you have to do is put that phosphate on and it's going to be active. So they work fast. Peptides work quickly. How long till they break down? Well, the half-life of peptides is not very long. We talked about insulin and how long that took. I had told you about 5 minutes. So what happens is that peptides must be secreted continually because they will have their effect quickly, but they break down quickly also. So they have to be secreted all the time. I'm going to stop there, and that's the end of the peptide hormones, and we'll begin with steroids on the next video. Thanks for checking in. Thanks for listening. Bye.